In 1990, the University of the Philippines established the Philippine Society of Microbiology, ratifying its bylaws and constitution. Its reach and to foster collaborative and is attended by speakers and check, 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 check. Microbiologists, 70 diplomates, and 27 fellows in its registry. Currently, PAM has been successful in endorsing the Microbiology Bill, which will regulate the practice of microbiology in the country. Another milestone in the history of PSM is the founding of the Microbiology Consortium, which aims to be of service by transforming microbiology education in the country by promoting new and more effective ways of teaching microbiology and excellence in ethics in the practice of microbiology as a profession by sharing resources and information among member institutions. The Microbiology Consortium was formally launched in 2018 during the 47th PSM Annual Convention through a ceremonial signing that included 31 HEI from different regions of the country. To recognize members who have excelled in microbiology and related fields, PSM has given various individual awards since 1985. The first recipients of the Most Distinguished Microbiologist Awards included Professor William Fernandez, Professor Macario Palo, Dr. Patrocinio Santos, Dr. Lydia Oson, and Dr. Ruben Aspiras. In 1987, PSM began the Outstanding Microbiologist Award to recognize exemplary members of the Society. As of 2019, 31 PSM members have been recognized for their outstanding achievements and contributions to microbiology research in the country. In addition to the Outstanding Microbiologist Award, PSM also recognizes educators for their exemplary teaching of microbiology through the William L. Fernandez Excellence in Teaching Microbiology Award, and institutions and individuals who have helped serve PSM in its activities and mission through the PSM Institutional and Individual Service Awards. Through the years, PSM has formed important linkages with the academe and industry. In 1994, Colgate-Palmolive Philippines Incorporated launched products in partnership with PSM. And in 1998, Procter & Gamble Philippines partnered with PSM, which continues until today. More recently, PSM has formed partnerships with other major sponsors like ESCO Philippines, 3M, and Defensil. And these partnerships have allowed PSM to carry out its various activities and support the research of students through the thesis subsidy and reproduction grants. Travel grants have also been given to deserving members. To increase awareness in microbiology and biotechnology, PSM also organizes various activities, in addition to its annual convention and cluster symposia. These include quiz contests for high school students, and more recently, caravans, career talks, and training workshops.
Good morning, everyone, and welcome back to the 52nd Annual Convention and Scientific Meeting of the Philippine Society for Microbiology. The theme, Microbiology for Unity and Prosperity through Collaborations and Industry Linkages. Day 2 activities include plenary lecture, 2 parallel technical sessions, and the closing ceremonies where we will welcome our new president and new board of directors for the next fiscal year. At this juncture, let me introduce the second speaker for our plenary lecture. Our second speaker is an associate professor of immunology at the University of South Carolina School of Medicine, Greenville, where she teaches immunology and virology to first-year medical students. She received her PhD in cell and molecular biology with an emphasis on microbiology, virology, and parasitology from the University of Pennsylvania, where she studied natural killer cell functions in the laboratory of Dr. Jordan Orange. She then received an Institutional Research and Academic Career Development Award postdoctoral fellowship to conduct research into the interferon-stimulated immune response to Sendai virus in the Department of Pathobiology at the University of Pennsylvania School of Veterinary Medicine while receiving training in undergraduate teaching.
Her research lab at the University of South Carolina School of Medicine, Greenville, is focused on host pathogen interactions during respiratory infections with a particular focus on intrinsic and innate immune responses. She also directs research programs to study infection susceptibility in healthcare providers and community health outreach needs and is committed to providing mentoring and research opportunities to undergraduate and medical students in Greenville, South Carolina. Without further ado, we would like to call on Dr. Jennifer Greer, Associate Professor of the University of South Carolina School of Medicine. Dr. Greer's presentation title is Salivary Surveillance of Antiviral Immunity in Emergency Healthcare Providers. Dr. Llewellyn Moron Espiritu, Division Representative for Medical Microbiology, will moderate the open forum after our speaker's presentation.
Good morning, everyone, and thank you for joining me. My name is Jennifer Greer, and I'm an Associate Professor of Immunology and Virology at the University of South Carolina School of Medicine, Greenville. And before I begin, I just want to take a moment to thank the organizers for giving me the opportunity to speak this morning. I'm really excited to share this work from my lab with the Philippine Society of Microbiology and all of you in attendance. And so my lab primarily looks at immune responses to respiratory infections, whether it's viral or microbial. And so with our association with the hospital, we got really interested in this project that sort of fell into our laps during the COVID-19 pandemic. And so today I'm going to be telling you a little bit more about salivary surveillance of antiviral immunity in emergency healthcare providers. And so, well, what I'm talking about today is primarily in the context of COVID-19 and infections with SARS-CoV-2, the virus that causes COVID-19. A lot of the concepts that I'm discussing are really applicable broadly to a wide variety of respiratory infections or even potentially emerging infections that we haven't necessarily learned about, but we may want to know some of these immune mechanisms. All right. So I'd like it to kind of take everyone on a step back in time to consider the early days of the pandemic as we were first learning about the infections as it started to spread around the world. And so the earliest cases that we're aware of so far occurred in December of 2019. And this was a novel respiratory infections that were reported in Wuhan, China. But then the infections continued to spread. And so here in the US, we actually reported our first case in January of 2020. And local to me in South Carolina, our first case was reported in March of 2020. And that's pretty much in line with the timing of the virus really starting to spread and go global and having significant number of cases in countries around the world. And so that sort of set it up where we have a global population that wants to know more about this virus and really wants to know how to prevent spread of the infection. But unfortunately, we weren't particularly well prepared for everything. And so we kept running into a lot of complications in these early days. One of the major issues that we ran into is because this was a novel infection, we just didn't have tests available. As things were being developed, we didn't necessarily have the supplies when we did know how to test for it. And so we were really limited in who could get tests, particularly in the US. There was also an issue of the limitations in personal protective equipment. So I remember N95s being very difficult to get, but recognizing that that was probably the best way to prevent spread of a respiratory infection. But for most individuals, getting hold of medical grade equipment or things that they could use to prevent infection was not necessarily readily available. And along with that, there was actually quite a bit of inconsistent information regarding how this virus is spread. And so in some cases, it went so far as to actually be misinformation. We also, in those early days, didn't really understand that there was a potential for asymptomatic transmission. So there's this potential that individuals who have no symptoms could actually be infected and still spreading the virus. We didn't realize that was a situation at the beginning, and that really kind of snowballed with the limitations on testing ability, so that in those early days, we were really only testing sick individuals or individuals with known contact. However, we didn't realize that you could have people that were quite healthy who were actually still at risk of being infected and even spreading the infection. And so that definitely made things a little more complicated. On the hospital side, there was also this situation where we had individuals coming in with really severe infections, but then also reports of individuals with infections with very mild symptoms or even asymptomatic infections. And so it really wasn't clear what was driving some of the variability there. And although we have a lot more information now, there was just a lot of unclear factors. And one thing that I was considering at that time as an immunologist is, was there the presence of potential existing immunity? So were there members of our population that might have had antibodies or components of their immune response that were able to detect and respond to SARS-CoV-2, even if they hadn't necessarily already been infected? And that thought sort of stems from the fact of the similarities between SARS-CoV-2 and some of the other coronaviruses, which includes the common cold coronaviruses. Present to the hospital for a COVID testing, usually due to severe symptoms, than non-essential workers. And if you break this down into different subpopulations of healthcare professionals versus support staff, still a very, very high risk. And even if you consider other potential socioeconomic or socio-demographic factors, that high risk exists sort of across the board within healthcare workers. That's not to say other essential workers were not at risk. They did also have a slightly elevated relative risk compared to non-essential workers. But for healthcare workers, it was just significantly higher. So that around that time, or shortly after, I found myself in a unique situation with one of my collaborators. So my collaborator, Lauren Fowler, who's now at Wake Forest, had actually begun collecting saliva samples from emergency healthcare workers as part of a separate study that she was conducting within the state of South Carolina. And that study had begun in September of 2019. And then she had continued to collect samples all the way through March of 2020, at which time it was deemed unsafe for her to be continuing to collect samples simply because of potential risk of infection. Within her sample population, she had collected salivary samples from emergency department physicians, from emergency medicine services technicians, or EMTs, as well as medical students who were working as EMTs. So here at the School of Medicine Greenville, all of our first-year medical students actually get EMT training, and then through their first 2 years, they actually do shifts on the ambulances as EMTs, so they get a chance to meet the community. And so we had samples from students that were working as EMTs. So altogether, my collaborator had collected roughly 160 salivary samples across 55 participants. And what was sort of interesting is that for every participant in her study, she had been collecting samples both pre and post shift. So that meant we had at least 2 independent samples for all individuals. And due to sort of the shutdown as things, in March of 2020, everything kind of closed down, all of these samples had been stored in the freezer. And so that meant they were still hopefully quite stable, I should say, for us to test and look at what information we might be able to gain from these samples. So we approached this project to sort of address some of those complications that we were seeing early in the pandemic. And so we wondered, by investigating these samples, could we get an idea of the presence of existing immunity in our community here in South Carolina? We also wanted to sort of understand, are emergency healthcare workers a good population to monitor spread? Recognizing that a wide swath of the population, regardless of background and demographics, usually ends up at the emergency room for any number of reasons. And so interacting with healthcare providers in the emergency room or through emergency transport gives us this opportunity to sort of get a snapshot of the community, particularly if we're thinking about infectious disease. And then the last question is, would these salivary samples provide another route for testing? Slightly easier to get, slightly does not require quite as much materials, and so and potentially quite stable. And so our thought was, is this now perhaps a way to increase test capacity in a time when other supplies are limited? All right, so this was our study population. We ended up with 9 emergency physicians, 7 Greenville County Emergency Medicine Services providers, or EMTs, and 39 students. And you can see the breakdown of male to female in those groups was somewhat similar, not a lot of variability. We did not get ages for emergency physicians. We do have it for the EMTs and the students, both relatively on the young side with some variability in the age. And then we do have information about the shift duration. So how long were they working between that pre and post shift? It wasn't something that we were necessarily focused on, but we felt it was worthwhile to have that information just in case. All right, so how did we approach this? First of all, I will say this was the winter of 2022. So all of these samples had actually been in storage for about 2 years at that point. So the information that we're gaining is going to be information gained after long-term storage, for better or worse. And we decided to start with post-shift samples. And that way we could do RNA extraction for qRT-PCR. That would give us an idea if there was any ongoing infections happening. So was the virus actually present? But then we could also do an ELISA for SARS-CoV-2 antibodies. And that will give us some information about prior infections. So has any of these individuals perhaps been infected previously, or potentially if they have had a previous infection that's given them some antibodies that might recognize SARS-CoV-2. And the kit that we used for the ELISA specifically looked at the nucleocapsid protein and the S1 receptor binding domain. And so that's important because we're thinking about what's going to be exposed particularly within the oral cavity where the salivary samples are being collected. So that seemed very relevant to us. And then for samples that did show up as potentially positive or maybe inconclusive, we then were able to repeat that post-shift test, so sort of a replicate of what we've already done. But then we made use of those pre-shift samples as a way to have sort of an independent sample from that same individual to kind of do a validation of the potential positives. All right. So if you're familiar with immunology, you may be thinking, why are you looking at IgG in the saliva when IgA is the primary antibody we think of as being secreted in the saliva? And so here's some data from Sabra Klein's lab at Johns Hopkins that really sort of shows that yes, serum IgA and salivary IgA do sort of correlate, but there's not a really strong correlation. And so if we wanted to use salivary antibodies as sort of a marker for overall protection and overall immune response, perhaps IgA isn't the best approach. If we look at IgG, regardless of kit and regardless of potential target, so whether we're looking at the N protein or the receptor binding domain, we actually see a really strong association with serum IgG relative to salivary IgG. So if we want to detect the presence of immunity, salivary IgG is going to be a really great way to do it. and thinking about, again, that receptor binding domain and having an antibody that might interfere with interactions of the virus with cells of the body, this is a really great way to go. So we thought salivary IgG would be an excellent way to look for this. There is also the added benefit that IgG is one of the more stable isotypes of antibodies. All right, so what did we find? Well, first of all, for our emergency physicians and our Greenville County EMS providers, we actually didn't see any reactive antibodies against SARS-CoV-2. However, in our student population, we actually did have 2 participants that tested positive for antibodies that were reactive against SARS-CoV-2. And you can see that the individuals were positive in both the pre-shift and the post-shift samples. So this is more than just one sample that showed up positive. We have a confirmation that it was in more than one sample from the same individual. And from a timing perspective, what's really interesting is positive participant 1. Their sample was actually collected in October of 2019, so far before we recognized any infections in South Carolina or even in the United States. Positive participant 2 was also earlier than those initial cases. And so this is really interesting and got us thinking, hey, what was going on here? And I don't know that we have an answer, but I will say this was a really exciting result for us. And so here we can just sort of show you how it matched up with the overall participants. So we had 2 students that were positive. We did not see any other individuals in the other groups. Both of our individuals that were positive actually happened to both be age 25. And that was interesting to us because some more recent studies have come out looking at serum antibody levels prior to the pandemic, and found that younger individuals, so those 17 to 20 or even younger, tended to be more likely to have antibodies that reacted to SARS-CoV-2 prior to infections in the area. And so this seems like it's quite in keeping with individual or what other studies are seeing within the serum as well. So we think the saliva IgG is actually proving to be a pretty good method of detection for this potential antiviral immunity. So as I said, this was really exciting because when we think about transmission of the virus, we know this is respiratory transmission. So a lot of air travels through our oral cavity. And so that's a place where virus, if it's either coming in or leaving the body, may come in contact with these potential antiviral antibodies. And so if the antibodies are present, there is the possibility that they can bind to the virus, neutralize it, prevent it from infecting the body, or help it from not infecting a next individual. And so we have this potential where if someone has either pre-existing antibodies or they've had some prior exposure from some infection or another, they may actually have some protection. We're not really certain how that protection will directly correlate according to the antibody levels that we're seeing. But it is exciting to think about that we can use this salivary detection method to get an idea of potential antiviral immunity. And so just to highlight the conclusions from this part of the talk, I just want to highlight that, so we were able to see SARS-CoV-2 reactive IgG, particularly in 2 students that were serving as EMTs. So that says that this immunity very likely existed prior to cases, or there is this potential that there is a potential for some cross-reactive antibodies. So either we had cases before we realized, or we have some potential reactive antibodies coming from an infection with a different coronavirus. Because of protections for our human subjects, we were not able to contact them for follow-up, so we don't know if they had a history of infection or exposure to someone who might have been ill, or a previous infection with a different coronavirus. So we can't necessarily narrow that down now, but regardless, we do think it's really exciting and interesting that we were able to detect the presence of these reactive antibodies in a salivary sample even 2 years after collection. So that is really important for us because it gives us some flexibility when we want to do sample testing, perhaps in the future or for emerging infections. But before I go any further, I wanna come back to this graph because this actually kind of led us down a bit of a side project. So what you may have noticed is there seems to be a pretty significant drop between pre-shift and post-shift antibody levels. And so that was really interesting to us and begged the question, does potentially stress impact salivary IgG? So during the shift, we would anticipate the students would be quite stressed. This is an EMT shift, can be dealing with traumas and individuals that are just, going through quite a lot medically. And so we thought perhaps stress was in some way impacting the presence of salivary antibodies. So we wanted to dig into this a little bit more. We know that there is a connection between stress and immunity. And so looking at some of the ways that this might kind of impact each other, we know there's some different types of stress. So examples are chronic environmental stress. Social situations at home, in your work environment. There's also physiological stress. So this is what we see potentially with sleep disturbances. This is quite relevant for our healthcare providers, particularly the emergency healthcare providers, because they are usually working at what's called shift work, so outside of the traditional 9 to 5. Work like that frequently leads to changes in circadian rhythms, disruptions in sleep, and obviously a very stressful situation in general. And then also considering that there are stressful events that can drive a stress response, and thinking about where our emergency healthcare providers work and what they were going through during the COVID-19 pandemic. This is probably a very valid source of stress that may have an effect on their immune responses. And so all of these types of stress feed into the brain, and then through a number of signaling pathways will lead to production of this chemical called cortisol. So cortisol, I'm not going to go too much into it, but we definitely know one of the mechanisms of cortisol, or one of the actions, is it is an inhibitor of the immune response. And so this is what kind of led us to this idea of, could stress during an emergency medicine shift have an effect on antibody production? All right, so that led us to sort of start a secondary study. And I will say the results from this study are very preliminary. We've just been getting the data in the last couple of weeks, but we had a number of participants, again, EMTs and medical students working as EMTs. Samples were collected during 2021, and this is important because some individuals were vaccinated, some were not, as vaccines were not necessarily available to everyone, and hadn't necessarily been required for our EMTs yet. And so we did that same salivary ELISA. This time we did it for both cortisol and for SARS-CoV-2 IgG. And within that population, we found 11 that were actually positive for the SARS-CoV-2 IgG. And interestingly and unfortunately, we didn't see that consistent relationship across a shift. So we didn't see a constant drop. But what is interesting is, if we sort of looked big picture, is we actually do see a decrease in antibodies as cortisol levels go up. So that does suggest that there is a relationship between the two, but perhaps not within that 12-hour window. To further support that, if we, and this is really interesting, is if we break out those positive samples based on occupation. So looking at our medical students versus our paramedics in terms of cortisol production, our EMTs had significantly higher cortisol production. And then on the other side, if we look at antibody production and the presence of salivary IgG against SARS-CoV-2, our EMTs actually had significantly less. So that again suggests that there's some sort of correlation there. Why might that be? Well, for sure, we're thinking about chronic stress here. So our EMTs, this is their full-time job. This is what they're doing day in and day out. And so that type of environment is definitely producing chronic environmental stress. And then you also have that physiological and stressful events just feeding right into that. So that clearly leads to increases in cortisol, very likely driving in some ways the decrease in salivary IgG. And we think we see this more in our EMTs because it is such a chronic situation, whereas our medical students were a little bit earlier in their medical career. and had just less EMT exposure. So this is a shorter period of time of super stressful time for them. But the question that remains to be seen is, how does this actually lead to disease susceptibility? And so I don't have an answer for you because, as I said, this is still quite preliminary. But we are hoping to get more information, more samples, and to get a better idea of, does this stress somehow lead directly through salivary IgG to contribute to disease susceptibility for our emergency healthcare workers? And I think if we can get more information about that, then gives us the ability to kind of go upstream and see if there's ways that we can change the stress environment and then hopefully give better protection to our healthcare workers. All right, so just some concluding points overall. So yes, we were able to detect antibodies against SARS-CoV-2. So that tells us this sort of banked salivary system is a feasible system for surveillance. I will say I didn't show you any of the viral RNA data because we couldn't make it work. And so that is one thing. If we want to set this up as a system for surveillance of virus itself, it will either need to be in terms of antigens, so that we're looking for antibodies against it or using ELISAs to detect those antigens, or we'll need some specific storage conditions for the viral RNA or whatever nucleic acid it might be. But then the other takeaway from this is that we think emergency healthcare workers really could be a good population for surveillance of outbreaks because of their interaction with the community, because of the variable risks with the stress as well. We think that there's still a lot to be learned there, but they really are a good population to sort of get a snapshot for what's going on in the community. All right. And so with that, I just want to take a moment to thank the members of my lab. I would like to just highlight Haley Meltzer here actually in the middle of our lab photo in the red dress, was the technician that did a lot of the saliva testing in those initial samples. She really was in the thick of it because we didn't know if they are infectious or not. So she did some absolutely fantastic work. And then DG Nieves Esparcia and Misbah Mukri are the students that have actually taken on the cortisol and salivary IgG project. And then all of my collaborators who have helped with sort of understanding stress, helping us with sample collection, connecting us with the emergency medicine departments at Prisma Health, and then also helping us with statistics and collecting samples. We would not have been able to do this without them. So thank you so much for your time, and I would be happy to take any questions that you have. Thank you so much.
Hello, my name is Daniel Figues. I'm professor at the University of
Didinig niya ba? Okay. All right. To our participants, do you have any questions? Who would like to start with the first question? Hi, Dr. Jennifer. Good morning from the Philippines. All right. Okay,
Good morning.
I think I'm audible. Okay. Naka-muted siya. I think Dr. Jennifer is muted. All right, to our participants, if you have any questions, kindly proceed to the nearest microphone. Okay, state your name and affiliation, please. Nadidinig. All right. Okay. From the other hall, because I can see the participants, baka may questions po. All right, maybe I'll start with the first question. I think, all right, okay. Hi, Dr. Jennifer. Thank you for that presentation. Okay, with your, I've noticed that your sample population, okay, initially was like 55, okay, a cohort of 55 people who participated. Okay, do you plan to upscale, okay, like do another surveillance and increase the population size? Because I was thinking, are we expecting the same, let's say based on statistics, if we have a larger population size? Thank you.
Yes, are you able to hear me now?
Dr. Jennifer is still muted.
I believe I'm unmuted on my end. Perhaps there's a sound. Is my sound coming through?
Ganito. O sige, opo tayo.
We can still hear Dr. Jennifer.
Ayan. Sige nga po.
I'm just testing to see if the sound will be loud enough for you to hear me.
We apologize for the technical difficulty. Our tech team is already working.
Yes, are you able to hear me now?
Right, yes, very clear. Thank you. Thank you very much.
Awesome. Wonderful. Yes, we do hope to expand our sample, particularly for understanding the stress and the salivary IgG. Unfortunately, we don't have any other samples from the really early time point in 2019 and 2020. So we've sort of exhausted all of the available samples to test that early surveillance. But we are still hoping, we can continue expanding the later study looking at stress and immunity. And especially now as more individuals are becoming vaccinated and we have more people that are willing to learn about their immunity, I think we'll have a much easier time recruiting participants to the study.
All right, thank you. Do we have other questions from our participants, attendees? Okay, ma'am. Okay.
Good morning, Dr. Gray. I am Socorro Paraco from Silangan University in Dumaguete City. I'm just, this is a basic question in immunology, maybe for you, very simple question. How much, what percent of the IgG in the body is present in the saliva?
So that's a great question. In terms of total IgG, I want to say it's about 5 to 10%. So serum IgG is definitely the largest component of IgG. But part of the reason we like the saliva testing is it usually has a pretty strong correlation with the serum IgG. Which has now we sort of understand is a good marker for protection against infection. And so we're hoping that we can use this sort of non-invasive, very easy way to get some information about quantities and relative amounts of IgG between individuals by collecting saliva samples instead of having to collect serum and blood, which is a lot more invasive. It requires a lot more materials and time and even a professional to do it. But yeah, great question. I should actually look a little more into that in terms of relative quantities. Thank you.
Okay, so I'm going to accommodate one more question. We have from Matthew.
Hello, good afternoon, Dr. Jennifer. So I'm Alana Gamboy from University of the Philippines, Manila. May I ask regarding the timing of the serum cortisol collection that you did in my experiments? Because serum cortisol levels also has variability throughout the day. So usually it peaks in the morning and throughout the day it declines. So maybe it also contributed to the correlation results that you saw with the reactive antibodies. So thank you.
Yeah, that's actually a really great question. Because we're looking at emergency healthcare providers, they're working shifts around the clock. And so that's one thing we do want to look at is in sort of the timing of their individual sort of circadian rhythms versus time during the day. So we do have that information and we're trying to kind of go back and do that evaluation, particularly for the later set. We do also, I didn't have a chance to show it, but we have data on empathy and sort of perceived stress. So how are they feeling burnt out? Because we think that may also affect stress levels as well, which then, as I showed, might translate into changes in immunity. So there's definitely a lot more really interesting questions that we think we can learn about from this data. Thank you.
All right. I hope that our audience of curiosity were met from the talk of Dr. Jennifer Greer. Now I will proceed with the awarding of the Certificate of Appreciation to our plenary speaker, Dr. Jennifer Greer. If you have the digital certificate, maybe you can flash it on screen. But let me read the citation. Right. The Philippine Society for Microbiology, INC, awards the Certificate of Appreciation to Dr. Jennifer Greer for having served as a plenary speaker with her talk, Salivary Surveillance of Antiviral Immunity in Emergency Healthcare Providers, during the 52nd Annual Convention and Scientific Meeting of the Philippine Society for Microbiology in cooperation with the Trinity University of Asia with the theme Microbiology for Unity and Prosperity through Collaborations and Industry Linkages. Given this 21st day of July 2023 at the Hilton Manila Hotel. Signed by our PSM President, Dr. Howard Ho. Thank you very much, Dr.
Thank you so much.
For a couple of seconds just for a photo of with the crowd here. Thank you very much. Just a reminder to all our members, KPSM members, you may cast your votes or ballots through the PSM PPP website or through the QR codes that are available on your tables. The voting process will be open until 1 p.m. 1 p.m. And for those who have visited all our exhibit booths for today, and completed the sponsor pass, please submit your sponsor pass to the Secretariat Committee. Baka po kayo ay mabubunot later for the raffle prizes. Thank you very much.
For the next part of the program, let me introduce our last plenary speaker for this event. Our third speaker is the founding director and professor in the School of Pharmaceutical Sciences at the University of Ottawa. He is a distinguished research chair at the University of Ottawa. He co-founded and co-directs the Shanghai Institute of Materia Medica, University of Ottawa Joint Center in Systems and Personalized Pharmacology. He was the founding director of the Ottawa Institute of Systems Biology. Prior to joining the University of Ottawa, he was the senior vice president at MDS Proteomics. Daniel obtained a BSc and an MSc in chemistry from the Université de Montréal.
Our third speaker obtained a PhD in chemistry from the University of Alberta under the supervision of Dr. Norman Dovici. He did his postdoctoral studies at the University of Washington in molecular biotechnology with Dr. Ruedi Aebersold. More recently, his laboratory has been developing technologies and bioinformatic tools to study the human microbiome. He recently co-founded a spin-off company called MedBiome, focused on precision targeting of the gut microbiome. With this, we would like to call on Dr. Daniel Figues, Founding Director and Professor at the School of Pharmaceutical Sciences of University of Ottawa. Dr. Tadeus Carvajal, the PSM Vice President, will moderate the open forum after the presentation of our speaker.
My name is Daniel Figues. I'm professor at the University of Ottawa, and today I'm going to talk to you about targeting the microbiome to improve human health. And this is an ongoing collaboration between the University of Ottawa and the Shanghai Institute of Materia Medica. What are you? This is a very important question. In fact, we are a symbiotic system composed of human cells as well as bacterial cells. And if you look within a typical person, they will have about 10 million bacterial genes. In terms of genomes, this is our part of the whole genomes in us, and those are the bacterial genes. So they play a very important role in our health and diseases. Now, if you look in terms of number of cells, about half of the cells, just over half of the cells in our body are in fact coming from bacteria. Now they are small, so they don't occupy a large volume. Nevertheless, they are present and they play a very important role. These bacteria are composed of about 1,000 different bacterial species. So this is a very complex system involving many bacterial species and the host itself. So it is truly a symbiotic system. For the longest, we've neglected what this that we were a symbiotic system. We've really focused on the drug development for the host, and in the last 10 years or so, we've realized that the microbiome is a very important player. Therefore, we need to understand its role and whether we can target the microbiome. Now, if we zoom in within the microbiome, and in particular the gut microbiome, reason for the gut microbiome, most of the bacteria, in fact, are present in the gut. That's the highest abundance of bacteria in human. And when you zoom in, you will see, in fact, that there are different components. As you are in the luminal part of the gut, there are bacteria that lives there and transit through the gut. And as you get closer to the host, you have a region which is called a mucus region, in which there's going to be bacteria that live within that mucus and are living there permanently. Now, as you get closer to the host, the mucus layer gets thicker and it becomes sterile, where therefore there are no bacteria that are in direct contact to the host under normal condition. This means that the interaction between the bacteria and the host are not physical. They have to be done through different means. And those means are through metabolites that the bacteria secrete and the host secrete, as well as protein glycans and microvesicles. The gut microbiome has been implicated in many diseases. There's been hundreds of publications over the last few years that have linked the gut microbiome to different diseases. And as we just explained, the gut microbiome does not physically interact with the host. So therefore, the interaction between the gut microbiome and the host, and in particular health and disease, has to be done through metabolites. Now, when we zoom in within the gut microbiome, we see that the top 100 bacteria represent 90% of the biomass. So they're going to be the bulk producers of metabolites, and they will be key to health and diseases for most instances. Now, the gut microbiome is in fact a represented pathway to precision microbiome therapeutic and nutrition. The microbiome is in fact a factory, manufactures many metabolites important to health. And the gut, for example, will produce metabolites such as dopamine, GABA, and tryptophan that either directly or indirectly impact the brain health. It will also produce metabolites such as TMA, which when oxidized generates TMAO, which is toxic to the heart, serotonin, and metabolites that will have an impact on the gut itself, such as butyrate and hydrogen sulfide. Now, there are many more metabolites that have been shown to be important that are produced by the gut microbiome to be important in health and diseases. Now, we would like to be able to take a drug and have this drug reach the gut microbiome to be able to have an impact on the metabolites that are produced by the microbiome. In this example, I'm showing you a drug that would eliminate the production of TMA and TMAO. You could have other drugs that would enhance or change the presence of specific metabolites. So there are currently no assay to test the effect of drugs on the microbiome and the production of metabolites. So how can we select a compound from a panel of compounds for the potential effect on the gut microbiome and its metabolites? Currently, this will be done by using animal model, which means that it's not amenable to a large panel of compounds, or using individual culture of bacteria. And we know very well that individual culture of bacteria do not represent the gut microbiome. So there's a challenge. Our solution to this is the RapidAIM technology. The RapidAIM technology allows us to take individual gut microbiome and maintain them in 96 well plate. Every well in the 96 well plate is going to be a living microbiome. And we have put in place techniques to isolate gut microbiomes and to maintain them in 96 well plate so that they will remain functionally in composition the same as the initial microbiome up to a few days. This then allows us to add compounds to those individual microbiome and have a readout that will tell us the effect of the compound on microbiome and the effect in terms of production of specific metabolites. Our technology has been validated through multiple publications. I'm just showing you a few of those. If you're interested, you can read on those background publications. I'm going to give you a few examples that relates to selection of compounds for the effect on the gut microbiome. And the first set of example I'm going to show you has to do with a panel of 40 drugs that are currently on the market for which we tested the effect against 5 individual microbiome using the RapidAIM assay. So basically 5 individual microbiome were put in 96 well plate and tested with 40 compounds. And we use this instance metaproteomics and our own software called MetaLab to read out the effects of those compounds on the microbiome. In this slide, I'm showing you the effect of the 40 FDA approved drugs in terms across 5 individual in terms of the biomass. Anything that is blue, the biomass in the well went down. Anything that is orange, the biomass in the well went up. So you can see the first 3 compounds are antibiotics, and as you expect, antibiotics will lead to a decrease in the biomass, and we observe a decrease in the biomass. This compound here is ibuprofen, and ibuprofen is an NSAID, and in all instances, ibuprofen decreased the biomass, and in particular in volunteer number 1, ibuprofen greatly decreased the biomass. And ibuprofen has been known to have a gastrointestinal effect if used long term and to deplete the microbiome. So this is consistent. This compound is methylprednisolone. Methylprednisolone in all of the microbiome increased the biomass, and in particular 3 out of the 5 microbiome, we saw a significant increase in the biomass. That means that when methylprednisolone reaches the gut, it's used by the microbiome and stimulate the growth of the microbiome, and portion of methylprednisolone therefore would not be available to the host. And we see basically that a set of compounds, about 20% of the compounds have effects in terms of biomass effects on the microbiome. And we see that about 80% of the compound did not have effects in terms of biomass, but also had effects in terms of a function of the microbiome. So the biomass did not change, but the function of the microbiome changed. You also see that some drugs, such as antibiotics, will have 100% penetrance, so they have effect on all the individual microbiome, whereas other drugs can have very specific effect in one particular subgroup of microbiome. Here, you might have noticed that for rifaximin, volunteer number 1, in fact, consistently had a growth in microbiome when treated with rifaximin, and we've repeated this experiment many times. And what we observe also in volunteer number 1 is an increase in antibiotic-resistant protein being expressed, whereas volunteer number 1 was not resistant to the 2 other antibiotics that were used. This is pretty interesting that we can pick up antibiotic resistance across a panel of healthy volunteers when treated with different drugs. Now you can zoom in on all of those compounds. Here I'm showing you the panel of NSAID and their effect on the gut microbiome. This is a controlled treatment of one individual, and as you can see, there's a stable biomass with different composition. When we treat with ibuprofen, you can see that the total biomass decreased. This is consistent with what we saw in the previous slide. And you can also see that the composition of the microbiome also changes. There are other NSAIDs that don't seem to have any effects in terms of the biomass. So the biomass is pretty similar. But when you zoom in, you will see that, in fact, the composition has changed drastically. So, for example, naproxen has a drastic effect in terms of the orange microbiome has disappeared. Similarly, for diclofenac, you can see that the bacteria from that group have also disappeared. So you can have no changes in the total biomass or no drastic effect in terms of the biomass, but you can have pretty important effect in terms of the composition of the microbiome by different compounds. So therefore, it's not sufficient to look at biomass as a readout of drug effects. You need to look at composition. And we're going to show you in a later slide that, in fact, you also need to look at function. And here I'm going to show you a few examples of different compounds and metabolites. The first example I'm showing you is a metabolite, a small protein called CLPB. And CLPB has been shown to be a metabolite secreted by the gut microbiome that goes into circulation and reaches the brain and is involved in control of satiety, so food intake. And we know that satiety is important in obesity, but it's also important in anorexia. And when we screen the panel of the approved drugs, we can find drugs that in fact will increase satiety and other drugs that have effect in terms of decreasing satiety across a panel of microbiome. So that means that this is interesting, and those drugs might be involved, might have secondary effect, in fact, in terms of weight control. The second example that I'm giving you is related to GABA. So GABA has been shown to decrease anxiety, stress, and depression. And GABA is regulated in the gut, and is an important source of GABA is coming from the gut microbiome. Here I'm showing you the effect of 40 drugs on the regulation of GABA. So those drugs in blue can increase the level of GABA, and the drug in orange decreases the level of GABA. Therefore, drugs can have also pretty significant effects on the production of GABA in the gut microbiome. The last example I'm going to give you from this panel of drugs is related to checkpoint inhibitor in colorectal cancer. So a few weeks ago, there was a paper that came out that shown, in fact, that metabolites produced by the gut microbiome, in particular inosine, hypoxanthine, and xanthine, modulate the response to checkpoint inhibitors in colorectal cancer. And here I'm showing you the regulation of inosine and hypoxanthine across a panel of those 40 drugs. You can see that some drugs have an effect in terms of significant effect in terms of upregulating the level of inosine or downregulating the level of inosine. The same for hypoxanthine. So they could have effects, or could potentially be also used to regulate those metabolites from the gut microbiome and have an effect in terms of the ability to use checkpoint inhibitors in colorectal cancers. Now I've showed you a panel of 40 different FDA-approved drugs. The next question we wanted to address is, can we see the differences between structurally similar compounds? So is the assay sensitive enough that we can pick subtle differences between compounds that are structurally similar? And to do this, we took another panel of 5 individual microbiome that were put in this 96-well plate, so living microbiome in this 96-well plate. And we would tested 20 structurally similar compounds on those microbiome, and we use as a readout 16S, metaproteomics, and metabolomics. Now, the 20 structurally similar compounds are structurally related to berberin, and I'm showing you the structure of the compound there, and I'm pointing 3 compounds: sanguinarine, ethoxysanguinarine, and dihydroxysanguinarine. So they have subtle differences in structure. We can see that they're all somewhat related to berberin, and those compounds were tested in our assay. Here I'm showing you some of the results in terms of the microbiome response to those different berberin derivative. So you can see, first of all, that every single individual microbiome illustrated by the square here are different, and that is expected. We expect that the microbiome between individuals are going to be different. And you can see in color in the circle around those the effect of the different compounds. You can already see that there are some compounds that are close to the control and some compounds that have different effects. Now, we use techniques to be able to realign the data, and what we see is, in fact, that there are some compounds that have effects between the treatment and the control, as you can see in those slides, and some other compounds that don't seem to have effects. Now, what we see is that berberins lead to functional changes in the microbiome. So here I'm showing you the functional profiles of those different drugs. And you can see that there are drugs that, in fact, have a very similar functional profile to the MSO. So that means that there's not much functional changes in the microbiome when we treat with those drugs. But as I pointed earlier, sanguinarine and ethoxysanguinarine have very strong changes in the functional profile of the microbiome. And you can see that some of the compounds, including berberins, have effects on the function of the microbiome. And you can really see it in this lower figure, where you can see figure B. We can see that there are very specific effects, sanguinarine in terms of cell wall, AEO sanguinarine, etc., defense mechanisms, signal transduction, replication, and so on. So those are not changes in biomass of the microbiome. They're really changes in terms of the function within the microbiome. And you can see that across a panel of structurally similar compounds, they do not all have the same effects. So therefore, the assay can be used. If we zoom in a little bit more, we'll see that, in fact, berberins can have effect, functional response at the bacterial level, in fact. Panel B and D tells you that there are functional changes, either up or down regulation in terms of specific group of function related to different genus of species. And if we zoom in panel C for 3 compounds, 13-methyl berberin, sanguinarine, and berberin, you can see that in 13-methyl berberin, that the bacteroides groups have the most changes in function, whereas for sanguinarine, the enterobacteria group have the most changes in functions. And enterobacteria are significant because this is a group of bacteria that have many pathogenic bacteria. So that might put a question mark on using sanguinarine. Berberin has effect on bacteroides and Akkermansia in terms of function. And interestingly, berberin has been used to lose weight by some individual. And Akkermansia is an important group of bacteria that have been implicated in the control of in obesity. Now, what we've learned using this panel of structurally similar compounds is that within a group of structurally similar compounds, they don't all have the same effect on the microbiome. And we can detect those effects all the way down to functional changes that occur within specific bacterial genus or species. It also tells you that the assay is sensitive enough to detect subtle changes between structurally similar compounds, and therefore could be used in preclinical selection of compounds that are of interest when one needs to select one compound versus other compounds in preclinical setting. so far, I've shown you example related to FDA-approved drugs, structurally similar compounds, and here I'm going to show you examples that relate to functionally similar compounds. Obviously, the function has been determined based on the effect on the host, and whether those functionally similar compounds will have similar effect on the microbiome is unknown. So we started again with a panel of microbiomes in the assay, and we tested 21 functionally similar compounds for the effects, and the readout was metaproteomics using our software. Here, I'm showing you the structure of the compounds, the names and their concentration that we tested them at. And the remaining set of compounds, all representing different structures, but similar function in terms of being artificial sweeteners. Now, most sugar substitute, in fact, through our assay, do not affect the level of bacteroides and firmicutes, and that's an important measurement in terms of effects on the microbiome, which is the ratio of bacteroides and firmicutes. And you can see that there are some, though, that seems to have effects on the bacteroides and firmicute, but most of them do not change the level of bacteroides and firmicutes, which is good news. Although most compounds did not have effect in terms of the level of bacteroides and firmicutes, when we zoom in terms of function within the microbiome, we can see some changes. For example, xylitol and isomalt have effects in terms of cell motility. You can see it, xylitol and isomalt. And in terms of coenzyme transport and metabolism, xylitol again and isomalt have strong effects on those specific functions within the microbiome. Other functions, such as extracellular structure, are being affected, in fact, by two compounds. MFE and REB have effects in terms of extracellular structure. And in terms of translation, ribosomal structure and biogenesis, you can see that the alcohol sugars have effects on those specific functions. So therefore, although they don't seem to have a very strong effect in terms of the bacteroides and firmicute level, they have effects in terms of specific functions that will have an impact on the metabolites that are being produced by the. So what have we learned so far? So we've learned that we can maintain living microbiome in 96 well plates, and we can test the effect of compounds on those microbiomes in terms of the composition and function of the microbiome, as well as the production of specific metabolites. We've also learned that FDA-approved drugs can have effect on the gut microbiome, and in terms of the composition and affect the metabolites and function of the microbiome. So therefore, drugs should be tested for the effect on the microbiome, even if they were not intended to target the microbiome, because those secondary effects on the microbiome could have long-term health effect for the host. Drug could also be repurposed to target the microbiome. So some of the drugs we've seen, and also screening the many FDA-approved drugs, we might be able to find drugs that can be repurposed to go to the gut microbiome to change either the composition, function, or metabolites produced by the microbiome. We've also seen that the Repligen technology is sensitive enough, is specific enough to be able to study structurally similar compounds. And we've seen that structurally similar compounds can have different effects on the microbiome. This means that the assay is suitable for preclinical development when you need to select compounds from a panel of compounds that would have either effect on the microbiome, or if you want to avoid compounds that have effect on the microbiome, because that's not your intended target. We'll also learn that functionally similar compounds can have different effects on the microbiome. So one cannot assume that because they have a similar functional effect on the host, that they will have a similar functional effect on the microbiome. In fact, they can have very drastic differences in their effect on the microbiome. So therefore, they should also be tested for their effect on the microbiome. What are the next steps? In our lab, we're currently doing a 1,000 compounds project across 6 microbiomes. And those compounds are selected such as drugs. We wanted to expand the number of drugs that we are testing. We also selected natural compounds. And the reason for that is that there are some very interesting diverse structure in natural compounds that are not available in conventional drugs. And therefore, we wanted to expand the number of the diversity of our structures. We also selected food additive that would potentially reach the gut microbiome, as well as agricultural compounds, because we know that many are residual in the food and therefore can have effect on the gut microbiome. We're also interested in putting in place an international consortium on microbiome compounds interaction. Reason being is there are many more compounds that are approved for usage, not just as drug, but for agricultural purpose, for the industrial application and so on. In fact, there's over 50,000 compounds that are approved for use, and some of those might end out being present in or affecting the gut microbiome, so therefore they should be tested. We think that the consortium could start with 10,000 compounds and in fact try multiple microbiome, healthy and disease, because we know that the response from healthy and disease microbiome to compounds can be different. We also want to develop the microbiome RX predictive. We've been developing machine learning and AI algorithm to be able to mine the microbiome drug interaction. And we think that as we populate our database, we'll be able to use the database and machine learning and AI tools to predict the effect of compound based on structural similarity. And we've already done proof of concept on this. In silico selection of compounds for screening, we're talking about a lot of compounds that could be screened. How do we select them instead of randomly testing compounds? Predict the on-off target effects on the microbiome. That could be interesting. Predict toxicity on the microbiome by looking at the functional changes that are present in the microbiome, and as well as stratification of patients based on personalized microbiome response. So we think that building this database, in fact, is going to be very important and will allow us to much better understand structure, function from compounds, as well as composition and function and metabolites from the microbiome. Now, this work would not be possible without a very strong team, illustrated in this figure, as well as collaborators from around the world. And you can see it in this interaction network of our collaboration network. We are funded by many different agencies to do this type of work. We've been very fortunate to get money from different pots. And if you're interested in studying the microbiome, you can check out our microbiome training program at this address. Thank you very much for your attention, and I hope to have a chance to interact at some point in the future.
So thank you very much, Dr. Daniel Fages. Very interesting research, and of course, this research actually focused more on the gut microbiome. Now, unfortunately, just this morning, Dr. Daniel Fages emailed and is currently traveling and will not be able to attend the Zoom meeting for our convention. However, if you have any questions about about the research that they do in the University of Ottawa, please try to email him. You can actually try to search him through Google Scholar, and his email address is available on his research website as well. Okay, so if you wish to have collaboration questions on methods, their laboratory and their constituents will be happy to answer. So thank you very much, everyone. I give the microphone to our moderator, Dr. Espiritu, for certain announcements.
Thank you, Dr. Paz. All right, again, we would like to remind all members of PSM to please cast your ballots. You may cast your ballots through the PSM 52 website or through the QR codes available on your tables. The voting process will be available and will be open until 1 p.m. To our participants, attendees, if you have finished already visiting all our exhibitors, our exhibit booths, you may please submit your sponsor pass, passport pass to our secretariat committee for the raffle draw that we will have this afternoon during our closing ceremony. Now we shall proceed to the next part of the program, which is the Microbiology Consortium of the Philippines meeting with member HEIs. To our attendance participants, we will continue with the viewing of the poster exhibits through online and on-site. You may also continue visiting the exhibitor booths. I think marami-rami pa po yung pwede natin puntahan dahil marami pa ang ating sponsors for this convention. We would like to invite everybody to our technical session this afternoon at 2 p.m. Okay, we have two technical sessions. This Hall A will be for the first technical session. The other technical session will be at Hall B. Okay? 2 p.m. this afternoon. Okay. For the member HEIs, may we request to please stay in this hall, Hall A. We shall proceed with the MCP meeting. So thank you po, and thank you everyone. Okay, enjoy viewing the posters and the exhibitor exhibits. Thank you.
After lunch, we will have the technical sessions at the afternoon. The sessions will be held in parallel here at the convention hall. For the on-site attendees, please return to the convention hall before 2 p.m. At this time, you may visit the exhibits and nominate the incoming board of directors. We hope to see you there.